Turn the microphone over to Dr. Stacy Ricci. And Dr. Ricci is in the Food and Drug Administration and the Therapeutic Biologic Biosimilar staff in the Office of New Drugs. Thank you.
Hello. Thank you, Dr. Lichtenstein, for that very nice introduction. You touched upon many of the topics that I'll go over. And it's good to emphasize and repeat them because they're complicated. Biosimilars represent a new licensure pathway for the FDA, and it's different from what the traditional drug development design that we think of. So just to briefly go over what I'll discuss, I'll give some of the statutory framework that the Biologics Price Competition and Innovation Act provided to the FDA for the licensure of biosimilars. I'll review terminology and also some of the general requirements that are needed in a biosimilar application submitted to the FDA. And also I'll review what the stepwise approach means to developing a biosimilar and FDA's recommendations as such, and also some unique development concepts that are specific for biosimilars and interchangeables. So the BPCI Act, or I'll just refer to it as the Act, was approved as part of the Affordable Care Act in 2010, and it created this abbreviated licensure pathway for biologic products that are shown to be biosimilar or interchangeable with an FDA-licensed reference product. So what's a reference product? That refers to the single biological product that was licensed under Section 351A of the Public Health Service Act. And the Public Health Service Act gives FDA the authority to license biologic products. An application that's submitted under Section 351A is considered a standalone application, and that would contain all the information and data necessary to demonstrate that the proposed product is safe, pure, and potent for use. And as Gary described, the statute contains these two requirements, these two criteria, in order for a product to be considered biosimilar. And that is that the product is shown to be highly similar to the reference product, notwithstanding minor differences in clinically inactive components. And so that would mean if there are formulation differences, for example, of the biosimilar compared to the reference product, and that there are no clinically meaningful differences between the two products in terms of safety, purity, and potency, or safety and effectiveness. Interchangeability means has three criteria. First is that it's biosimilar, and second, that it can be expected to produce the same clinical result as the reference product in any given patient, and that for a product that is administered more than once to a patient, in terms of safety, the risk in terms of safety or diminished efficacy of alternating or switching between use of the product and the reference product back and forth is not greater than using the reference product alone. And so what this means in practice is that an interchangeable product may be substituted for the reference product without the intervention of the healthcare provider, so at the pharmacy level. What's required for a 351K application or a biosimilar application? The applicant will submit, among other things, comparative data comparing their biosimilar product with the reference product, both analytically and clinical data, as well as publicly available information regarding FDA's previous determination that the reference product is safe and effective. And this licensure pathway is based on, in comparison to the 351A standalone pathway, less than a full complement of product-specific preclinical and clinical data. And this is why it's called abbreviated. So what does this mean, abbreviated? It does not mean less than in terms of any information that's required to support its safe, pure, and potent. And it's a different standard that's applied to the biosimilars. And so the data package that's required for a biosimilar or interchangeable product is very extensive. And it includes analytical data, comparative analytical data, non-clinical data, and clinical data. And then once a biosimilar or interchangeable has been approved, healthcare providers can expect it to be just as safe and effective as the reference product that it was compared to. Okay, so let's go over some of the information that's needed for the application. And so the point that must be met is that the product is considered biosimilar, and that also that it's clearly described that it utilizes the same mechanism or mechanisms of action for the proposed condition of use, meaning indication or dosing regimen, but only to the extent that the mechanisms are known for the reference product. And the conditions of use and the proposed labeling for the biosimilar have been previously approved for the reference product. It has the same route of administration, dosage form, and strength as the reference product, and is manufactured, processed, packed, or held in a facility that meets the same standards designed to assure that the biologic product continues to be safe, pure, and potent. The 351K application must also include information demonstrating biosimilarity based on data derived from analytical studies demonstrating the product is highly similar, animal studies including the assessment of toxicity, and a clinical study or studies including the assessment of immunogenicity and pharmacokinetics or pharmacodynamics that are sufficient to demonstrate safety, purity, and potency in one or more conditions of use for which the reference product is licensed. So this does not mean that each of these data elements are required. FDA can determine that one of the data elements is not relevant for the product or is not needed. So what are some key development concepts that differentiate biosimilars from standalone development? So let me just make sure I hit all the points I wanted to do. So the goals are different. And so on the left, this is the traditional drug development concept that people are familiar with, where you collect analytical data, non-clinical data, clinical pharmacology data, and you proceed through the safety and dose range finding clinical studies until, if hopefully the product appears to be efficacious, and one or more phase 3 studies are conducted in the given indication in which licensure is sought. In contrast, for the 351K pathway, the goal is to demonstrate biosimilarity or interchangeability, and it's not to independently establish the safety and efficacy of the product. And so the abbreviated pathway means that the product can be approved on less than a full complement of product-specific preclinical and clinical data because the biosimilar sponsor is relying on information that's already known about the reference product. And so this approach avoids unnecessary, expensive, and unethical duplications of studies and allows safe and effective products to be made available to patients faster and at a potentially lower cost. And as this figure is meant to convey, while the studies needed for product development overlap, the relative emphasis on each data element is different between the two pathways. A second key concept that the FDA recommends is that a stepwise approach is taken to generating data in support of demonstration of biosimilarity, where first the stepwise approach begins with comparative analytical studies. And the sponsor should then consider the role of animal studies if they are needed for their product, and then move on to conduct the comparative clinical PK and PD studies, including an assessment of immunogenicity, and then at each step, the uncertainty about what's known between the similarity between the two products would dictate the design of the studies that then follow. So if following the clinical PK, PD, and immunogenicity assessments, if no further information would be needed, then a comparative clinical study in a patient population to demonstrate equivalence efficacy may not be needed. And this is what's meant by the totality of the evidence to support the demonstration of biosimilarity. And again, the above list does not mean that every type of data described is necessary for a given biosimilar. A third key concept is that the analytical similarity data form the foundation of the biosimilar development program. That's why it's like the biggest box at the bottom of the pyramid. There's extensive physicochemical and functional characterization comparing the biosimilar product to the reference product. And to do this, multiple lots of the biosimilar product are generated and tested, as well as even more lots of the reference product are tested. And as you can imagine, there's all these different analyses are run, not only on the primary sequence being identical, which is part of the requirement, but that there's the secondary and tertiary structure of the protein molecules are highly similar, that the thermal and temporal stability of the biosimilar product is highly similar to the reference product, and that most importantly, that the biologic activity, the evaluation of the attributes that indicate the mechanisms of action for the product are similar. And usually the tests that are run for that, there's more than one that would evaluate the same mechanism, and so on. So, and then a fourth key concept is that the role of clinical studies differs from the standalone, for a biosimilar differs from the standalone program. And the nature and the scope of the studies will depend on the extent of residual uncertainty about the biosimilarity of the two products after conducting this extensive analytical analysis. And the types of clinical data that the FDA seeks are first based in similar PK and where relevant, similar PD, because these are considered the most sensitive clinical endpoints for assessing differences between the products, should they exist. Typically, the PK measurements involve area under the curve calculations and Cmax comparisons in a sensitive population. It may be healthy volunteers, but it may not be ethical to conduct the study in healthy volunteers, in which case a patient population would be used for that evaluation. And if the PD biomarker is available, then it would reflect a known biologic effect of the drug. The PK and PD similarity data support the demonstration based on the assumption that the similar exposure of the biosimilar to the reference product would be representative of similar safety and efficacy. Establishing that there are no clinically meaningful differences in the immune response between the proposed biosimilar and the reference product is also a key element in the demonstration of biosimilarity. And in certain cases, as I said, an evaluation of PK, PD, and immunogenicity may be sufficient to support the clinical criteria of that there being no clinically meaningful differences between the biosimilar and the reference product. Otherwise, then a comparative clinical study is conducted in a patient population that compares the biosimilar to the reference product. And the study design features, including the study population, the endpoints, sample size, and study duration, should be adequately sensitive to detect differences between the two products, should they exist. Okay, and the final concept that I will discuss is extrapolation, which Gary touched on, and this is a difficult one for a lot of clinicians, especially. So the potential exists for the biosimilar product to be approved for one or more condition of use for which the reference product is licensed, based on extrapolating data that's intended to support the demonstration of biosimilarity from one condition of use to other conditions of use, meaning the biosimilar development program would not need to have a clinical study in every indication in order to be licensed to treat those indications. In order to do this, sufficient scientific justification for extrapolating the data from the biosimilar product, from the reference product to the biosimilar product is necessary. And FDA guidance outlines the factors to consider to support this justification. And these include what are the known and potential mechanisms of action in each indication? What are the PK and biodistribution in the different patient populations? Are there immunogenicity differences in different patient populations? Are there differences in expected toxicity in each condition of use and patient population? Or are there any other factors that could affect the safety or efficacy of the product in each condition of use, which, for which licensure is being sought? So if there are differences, these have to be analyzed for the biosimilar product, and all the features that would impact those factors would need to be described. So to provide another cartoon to explain this, again, for standalone drug development, this is the typical clinical progression for collecting information in order to support the first indication that the product receives a license for, the second, third, fourth. Phase 3 efficacy, safety and efficacy studies are done. For biosimilar, this is different. So the goal of the biosimilar is to show that the product is both highly similar, notwithstanding highly similar from the analytical and PK perspective, and that there are no clinically meaningful differences between the two products. And once this demonstration of biosimilarity has been achieved, then the data from the reference product can be used to support the additional indications for the biosimilar product based on the evaluation of those factors that we outlined already. So in summary, I hope that these concepts have been laid out a little more clearly of your understanding of biosimilarity. And so the development of a biosimilar product is different from the standalone product development. And the development goal is not to reestablish safety and effectiveness, but to demonstrate the product is highly similar to the reference product and that there are no clinically meaningful differences. The analytical comparisons between the two products are the foundation for determining whether products are highly similar. Clinical PK and or PD, if relevant, is generally considered the most sensitive endpoint for detecting differences between products. An assessment of immunogenicity is needed, and a comparative clinical study is conducted if residual uncertainty remains about the biosimilarity of the products. The biosimilar is approved based on the totality of evidence that's submitted by the applicant, and they are approved based on the same high standards as for other therapies, and they are safe and effective when used for their labeled indications. So there have been 5 biosimilar products approved by the FDA, the first being Zarzio in March of 2015, the most recent being Renflexis that was approved just last month. And then some useful links, which I realize are going to be too long for you to write down, but there's, if you go to the FDA.gov website, there's a lot of information about biosimilars. There's a CME course, online course that you can take, and if you just search for it, you can find it. There's 8 available guidance documents from the FDA about biosimilars, guidance for industry documents. And also, it might be of interest for people who want to learn more about the concepts and how they have been publicly described by looking at the transcripts or by viewing the advisory committees meetings that have occurred for the products, including the 2 for the infliximab and the adalimumab products would be under the Arthritis Advisory Committee meeting web page. Okay, with that.
Thank you.
Thank you.